Good morning, everybody, and welcome to this, what I hope will be an interesting session. I think Brian Lewis, in his introductions to this whole meeting, really laid out some of the issues we're going to get into in this session about sustaining funding and progress in kidney cancer. And I'm going to hopefully, in 5 minutes or so, go through some of the data behind some of those issues, and then leave it to my colleagues here on the stage to discuss some of the research that they've done in the programs that they've led that have led to the progress we've currently have, and then we can have a discussion about how we can sustain that. So let me move forward to my slides. And I'm talking about some of the historical funding landscape from the NCI, the DoD, foundations and advocacy, industry, biotech, and some of the other organizations such as ASCO and AACR, and try to give you some of the data. So one of the things we've seen is that from the NCI, kidney cancer research funding has plateaued since 2022, while funding for other diseases such as breast cancer and lung cancer have continued to grow. Right now, we're plateaued at about $87.7 million per year, which represents about 1.2% of the total NCI budget. If you look at the amount of money given to kidney cancer over the last 5 years, about a little over a third of it goes to intramural program at the NCI, while other programs such as R01 funding and U01s, R35s, R44s make up the bulk of additional funding. And then there's funding for P30s, the Cancer Center grants, and the 2 SPORE grants, and a little bit for early career, and very little in PO1 support. And if you break that down by years, you can see that see if this pointer works, you can see that the funding has gone down slightly, and most of that decline has been in intramural funding. It's interesting that despite the attempt to cut 40% from the NCI budget overall, the amount of funding, thanks to efforts from Congress, has remained about the same for the extramural program. But in 2026, the Office of Management and Budget has established new rules which have inserted political review into funding decisions, restricted international collaborations, and increased some of the administrative burden related to grant funding. And it remains to be seen how that's going to impact the research that comes out and is supported by the NCI. We've also benefited, and thanks to organizations like KidneyCAN for lobbying for this, in the past, to a lot of support from the Department of Defense, with about $50 million allocated between 2022, or annually from 2022 to 2024, divided up, as you can see here, with the majority of that funding going to IDEA awards and concept awards. And we'll hear a little bit from Dr. Jonash about the Kidney Cancer Consortium, and a little bit from Dr. Abhishek about the Academy in their discussions. But you can see in 2025, $0 were allocated, and thanks to some lobbying efforts, it's been restored to $15 million per year in 2026. And this has created a big threat to basic science and translational research. The advocacy and foundations have been helpful in providing research support. We can see how that's broken down from the Kidney Cancer Association, KCRS26, KidneyCAN, and then also ASCO and AACR, but altogether this amounts to less than $1 million of support annually. So if we add all that up, you can see how it breaks down, with the majority of support coming from the NCI, some support coming from the DoD, and some support from foundations. But thanks to a lot of really great basic science work funded through these other mechanisms that have led to 2 Nobel Prizes, one in HIF and one in checkpoint inhibitors, there's been a lot of development at the pharmaceutical level, and we estimate, you can't see this well, but that about $1,200 million have been invested over the last few years in terms of developing various drugs that have helped to increase the median overall survival for patients with metastatic kidney cancer from 13 months in the interferon days to 17 months with IL-2 to now close to 5 years. And that's been great for our patients. Participating in those studies has been great for our academic investigators. And as you can see from this slide, where the greatest increase in support for clinical therapy over the 2015 to 2020 was for kidney cancer, with an 18% increase due to the development of therapies that have extended survival. So those included 17 new drugs and drug regimens that were approved, 5 first-line IO combinations, 2 adjuvant regimens, new second-line options, which we'll hear about from our other speakers, Nobel Prizes, as I mentioned, in HIF and in checkpoint inhibitors, have led to biotech picking these up, and then ultimately industry picking these up and turning these into drugs. And there are several new approaches that are under investigation. You'll hear about these during the meeting, but the bar is higher given all the approved therapies to get these new approaches into new drugs, and it remains to be seen whether biotech and industry will continue that level of investment. So, as we launch this session, how are we going to support sustained progress in kidney cancer? And you'll hear from brief presentations one after another from the speakers up here, and then we'll have a brief discussion about opportunities for future investments and support. So I'm going to bring up Abhishek Chabarty from the Cleveland Clinic, who's going to talk about his experience with the AKCI. Here we go, sorry.
So hi folks, can you hear me? When I got invited to do this thing, I wondered why I was doing it, and then I realized that I'm a bit of a symbolism for this session. So right now we are constantly making decisions about what's desirable and what's accessible. So we desired Brian Reaney, but you end up with me. So these are, you have lean times, you make tough decisions and make do with what's available, yeah? So I'm going to talk to you about the Academy of Kidney Cancer Investigators. We are now in our second cycle. This academy got funded for the first time in the 2019 cycle. The money kicked in 2020. And honestly, if there is one silver lining, it is that this came up for renewal just 1 year before the KCRP program was zeroed out. So we snuck in the renewal, We thankfully got good scores where the renewal did happen, and the money now is okay for the next 5 years. This is what the leadership structure currently looks like. Brian and Jim Roguroulas are still leading this academy. Brian Rini is doing incredible amount of work to keep this effort going and functioning. But as you can see, we've brought in some new folks. Many of the people on the dash today, as well as in the audience, started as advisory board members, but we now have new folks who have stepped in as the next cycle has begun. We have also very actively ensured that we have participation from patients, patient advocates, to ensure that their voice is being constantly heard. Surveys are being done to ask them what their needs are, and we constantly incorporate those ideas into how projects are developed by the academy. This is what the academy now looks like. So the first 2 cohorts have now graduated. The third one will be graduating this fall. We are spread out pretty widely across the entire country right now. In fact, I don't know if, I think I saw Maxine earlier. She's, I think, our out-of-country representative as well, because I think she has like a Montreal footprint as well. As you will see, we are spread out in terms of early investigators across the US, as well as one location in Canada right now. Most of the folks, I will say, based on anecdotal experiences as well as conversations, have found this network incredibly fruitful in our careers, as we have launched and started to get ideas and mentorship from real experts and as well as patients to see how we gear our own research and our own ideas over here. So we had set up some very clear initial as well as ongoing milestones, both in terms of funding as well as manuscripts and practice-changing discoveries that the academy would make, and how we would be providing mentorship to all the early career investigators as we went along. We're giving you very small examples of how we have done. And overall, I will say that all the metrics that we had initially proposed, we would be able to meet. We have, in general, exceeded those expectations. The initial funding that was provided by the academy has been leveraged by many investigators in the academy now to get follow-up federal grants, which are pretty large in size. So the ROI has been pretty good. The ROI on even manuscripts as well as practice-changing guidelines has also been pretty remarkable. Many of us have been able to launch careers during really bad time, like the pandemic had shut down many of our labs when we first started. But despite this adversity, we have been able to establish, I would say, fairly productive research programs. David is here, Ritesh, Vincent is around somewhere. Many people that you constantly think about in terms of how you're thinking about your general practice. All of these folks have been very active members of our academy and are constantly helping move this effort forward. So we have 3 career projects right now. One is geared towards physician scientists. The other one is geared towards more basic researchers like myself. And then there's a third one where we are taking a lot more community-centered efforts over here. And any idea that you might have, any feedback that you can provide to us to try to improve these areas would be really helpful. So one plug I will make, for example, is anytime, this is just fact, historically, anytime when funding becomes limited, the entire pay line almost always shifts towards more application, more translational projects, always. Because the people who are reviewing always want to see immediate, tangible benefits a month from now, 2 months from now, 3 months. Like, what am I going to do to my practice on Monday when I go back? This is not how discoveries really work, though. I cannot tell you how my work today is going to impact your clinical practice on Monday. If we would have done that, we would not have had checkpoint inhibitors, we would not have had HIF, we would not have had any of the remarkable things that we talk about. We wouldn't have had CRISPR. So we have to, as a community, not succumb to this temptation to only pay out the most immediate, tangible benefit, but rather have the appetite to continue to sustain high-risk, innovative ideas that perhaps 10 years from now, maybe one of them will change the face of how we are doing practice. So it's necessary to balance these 2 aspects, and I'll just stop over there, and I'm happy to take additional questions during the discussion.
So next, Eric Jonas is going to talk about the Kidney Cancer Consortium.
Good Good morning, great to be here. Amazing presentations already so far. And what I'm going to talk about this morning is the Kidney Cancer Research Consortium. So what is the KCRC? This is a DoD-funded multi-institutional infrastructure exercise, creating infrastructure that allows us to perform clinical trials that wouldn't otherwise be performed at a single center level to answer key scientific questions. You can see the individuals on the slide here are the key investigators. We have 7 institutions that really form the core of this. We also have satellite sort of partner institutions that work together with us in an ad hoc fashion. We have an amazing external advisory board group coming from academia, from pharmacy, from informatics that really help guide us in how we do this. And basically what MD Anderson does here is provide an infrastructure that allows us to perform these trials, trial design, trial statistics, as well as also then trial monitoring. So the mission really of this organization is to perform, like I said, the studies that otherwise would be difficult to perform in a pure pharma sort of supported manner. And by having this infrastructure, it allows us in an iterative fashion to gather information from each of these trials to really generate sort of a database of knowledge that we're constantly building on. The trials that we are doing or have done already really follow some key themes: immunotherapy modulation, rare tumors, key sort of areas of disease states. And for each of these trials, we're working very hard on having sort of a common underpinning of biomarker acquisition, also quality of life instrument acquisition. So once again, the totality of the information that we gather over time is going to exceed the individual parts. You can see some of the trials here that we've already completed have been IO-based, sort of IO+. We have several of these other trials that are currently underway that are really designed in a very similar manner that it's going to allow us to perform this cross-trial comparisons. And we're also moving into looking at how does HIF-2 alpha inhibition modulate the tumor microenvironment in the neoadjuvant setting, and is HIF-2 alpha inhibition an important way of treating individuals with very good risk disease. So this really allows us to sort of think mechanistically and thinking about the tumor biology and how these various agents that we are applying in these clinical trials interact with various aspects of tumor biology, really to refine mechanistic hypotheses. From an infrastructure perspective as well, by having these amazing institutions, you're going to be hearing about the SPOREs at some of the partner institutions that we're working with, we have this aggregation of information as well as expertise that goes beyond the clinical trials that allows us to really draw on these centers of excellence. And by doing that, we then have these various domains of interest and domains of expertise that are being led by various members of the KCRC that allow us to really increase the value of the work that we're doing with the clinical trials. So we started off modestly now more than 5 years ago. Our first trial was a biomarker study. We've now launched and have completed multiple clinical trials. As you see, we have multiple clinical trials that are currently underway. And really at this point in time, we're sort of in the small IIT level sort of clinical trial development phase. We anticipate moving this further into biomarker development as well as moving into earlier phase novel agents. And we expect that in the next 5 to 10 years, we're really going to be able to then start targeting these niche treatment areas, for example, rarer tumors and that, where a single institution would never be able to do this. We're going to have the scientific knowledge as well as this infrastructure that allows us to perform these trials in ways that would not be otherwise possible if you did not have the KCRC. So thank you for listening and look forward to our conversation.
So next we have James Brugerolis from the UT Southwestern is going to talk about some of the great work that's done in their kidney cancer spore.
So good morning, everybody. So it's a pleasure to be here. So I was asked to talk about the impact of the sport at UT Southwestern. This is a relatively young sport. We're still within our first decade. I believe the Farber sport is already into maybe their third decade. All right, so maybe a round of applause for that. So our goal is to proceed from discovery to translation, sharing, and scaling up. So this is perhaps one of the highlights of the kidney cancer program, going from the discovery of the HIF-2α protein to the development of a first-in-class drug, which is now approved by the FDA. You're all familiar with this journey. HIF-2α was discovered at UT Southwestern by Steve McKnight and David Russell. Structural studies led to identification of an unusual cavity. There was a screen of our chemical library of 200,000 compounds, which led to identification of small molecules. McKnight founded Peloton Therapeutics across the street. Peloton developed several analogs. We tested them in Bill Kellen's lab as well in preclinical models, providing evidence of activity against kidney cancer. Kevin Cornell at UT Southwestern led a multi-institutional phase 1 trial that showed safety and promising activity. And eventually Peloton developed PT2977, which we all know as belzutifan, and is approved today by for 4 different indications. So this is an unusual journey where an academic institution, a spin-off company, we went from gene discovery, protein characterization, screening to a drug that is now FDA approved for multiple indications. We've building upon pioneering clinical innovation around the stereotactic radiation. We have built a leading early phase investigator clinical trial platform using stereotactic radiation. We were one of the 2 institutions that pioneered the use of stereotactic radiation for oligometastatic RCC, along with MD Anderson, for oligoprogressive RCC, along with the Canadian Consortium. Along with many institutions, we've evaluated stereotactic radiation for primary tumors. I believe we first reported the use of stereotactic radiation for tumor thrombi, assemble a global experience, and we have a clinical trial looking at stereotactic radiation surgically. And I wanted to include this here because even though we have not done a clinical trial for brain metastases, we were one of the early adopters of radiosurgery. In fact, you can see here in 2019, we reported the use of stereotactic radiation for greater than 25 brain metastases, but we did not think that there was equipoise where we could compare radiosurgery to whole brain radiation, even with hippocampus sparing or memantine. We've also been involved in defining mechanisms of resistance. So we reported identification of 2 mutations that are associated with resistance to HIF-2 inhibitors, one in HIF-2α, the other one in HIF-1 beta. And then we provided some insight into a long-standing paradox, which is why is it that rapalogs are not associated with target mutations? And our data suggests that these drugs act primarily on the tumor microenvironment rather than the tumor. The program has made some foundational genetic discoveries, including the fact that VHL is insufficient for clear cell renal cell carcinoma. We discovered BAP1, found that it was associated with high-grade inflammation and poor outcomes, along with others, including Ari's work. Identified, I believe, the first mutations in mTOR in clear cell, provided the foundation for a molecular genetic classification. We implicated MITF in TRCC. I believe we first discovered NF2 mutation associated with non-clear cell renal cell carcinoma and identified that BAP1 germline mutation can be associated with a novel familial kidney cancer syndrome in collaboration with Maston Linehan. We have a very large ecosystem involving more than 50 laboratories. I could not list them all in one slide. I have here Bruce Beutler, who got the Nobel Prize for TLR for his work on toll-like receptors. And there are some people that are attending the conference here, including Mara, who I believe is one of the awardees, and Ching Sang. So a lot of labs doing impactful research. You can look at some of these publications in high-impact journals. We have also made a big investment in developing resources, not just for us, but also for the community as a whole, and have shared 661 samples globally. We have a tumor bank of live frozen samples that involves more than 2,000 patients. We've generated tumor grafts after implanting more than 2,000 patient tumors in mice, which we have distributed very broadly, primary cell lines, genomic data sets, genetically engineered mouse models for clear cell, sarcomatoid, as well as translocation. And then you heard from one of the earlier speakers about some of our work on digital pathology. So it takes a village. We are delighted to have a very large community. You can see here the leaders of the different SPORE projects. This was a picture. The quality is not very good, but this was our 10th anniversary faculty symposium a couple of years ago. And we are also delighted to have an outstanding team of clinicians and clinical investigators, including Hans Hammers and Tiang Sang, among others. Thank you for your attention.
And finally, we're going to hear from David McDermott from Beth Israel Deaconess Medical Center, Dana-Farber Harvard Cancer Center, talking about the Harvard Cancer Center kidney cancer SPORE.
Thanks to the organizers. I'm here to talk about the SPORE that Mike built. And these are sort of the three themes of my talk, which I'll cover briefly. Many of them are similar to what Dr. Brugarolas just talked about how the SPORE is so central to our kidney cancer research program. And I think that what we've learned about research over the last 25 years through the SPOREs is sort of, I think, gives us some hope of where we can go in the future as a field, despite the challenges with funding. The SPORE sits in our kidney cancer program at the Harvard Cancer Consortium here in Boston, and here are the leaders that make the SPORE work. What you notice is they work at multiple different institutions. They often are from multiple different disciplines, and that's sort of the strength of the program. But probably the most important person in the program is here with us today. That's Sabina Signoretti, who is our translational pathologist. If you don't have a Sabina in your research program, you need to get one, because she essentially connects our discovery science that Abhishek was talking about, which is so important, with our clinical impact through her annotated tissue bank, and more importantly, how she helps keep us on the rails when it comes to doing high impact research. She essentially is designing many of our trials and making sure they are well executed. From the translational side. The SPORE, as I mentioned, has been around for a while, almost 25 years now, as Jim mentioned. Mike was the original program director in 2002, and this is the list of the 5 research programs that existed at that time, and it's amazing when you go back and read some of these, which I did, how much progress has been made by all of you over the last 25 years. Just think of all the things we didn't have 25 years ago, like the internet and RNA-seq and all that sort of stuff. But in that time, we've benefited as a research program from the scientists that we've gotten to bring in or recruit into the program over the years, and this is just a few of them. And essentially, we've been able to take some of their discoveries along with all of you and turn them into clinically impactful therapies, as been mentioned. A lot of this award-winning science, has made a dramatic impact on outcomes. This is just 2 of the more important people within our program, Arlene Sharp and Gordon Freeman, and their labs have moved both in and out of the SPORE over the years, which is an important aspect of the SPOREs, is that they can be modified as you go along, as the science takes you in exciting directions. When we think about going forward, we're learning from what they've taught us and modifying our projects accordingly, and we hope to start another 5-year cycle of funding this September. Still waiting on that NOGA, though. As I mentioned, the SPORE is the hub for research, but it's not the only research that's going on within the kidney cancer program. But without it, all of this other research would not be possible. All of the clinical trials would be less impactful. The number of publications would probably be fewer. and the number of grants that are coming off having a SPORE would also be less. You see some of the recent R grants that have been funded within our universe, or all are helped by having a SPORE to collaborate with. I could point to a couple of different examples, but I'll just focus one, given on limits on time, our collaboration with IARC, which is located in Lyon, France. And this goes around the KIM-1 story, which to me is one of the most impressive stories within the SPORE, and this started over 20 years ago, and it started outside of the SPORE in nephrology with folks like Joe Bonventry and others working on kidney injury molecule 1, and was brought in with a developmental research project with the help of folks like Rupal Bhatt and Mike Atkins, and now Vincent taking the lead here, you know, and then collaborating initially with the folks at IARC, looking at whether KIM-1 was elevated before patients actually were diagnosed with kidney cancer, and in that one small investigation, they actually found that it was. Subsequently, we've gone to look at KIM-1 in multiple different stages of kidney cancer, but none of this would have happened had there not been the magnet to bring scientists from outside the kidney cancer world into the world in which we live. Probably, and lastly, I would say the thing we're most proud of as a program is our career enhancement, bringing in junior faculty who may not have originally thought of focusing on kidney cancer into the field. These are just some of the people who've gotten career enhancement funding over the years. Many of these folks have gone on to be national leaders and also international leaders, and along with all of you have made an ongoing impact in the outcomes of our patients, and we certainly hope and expect that to continue going forward. Thank you for your attention. I look forward to your questions.
So first, I want to thank all the speakers for sticking to the brief time constraints so that we'll have time for discussion and for your excellent presentations. I'd like to invite Tony Shueri up to fill the last chair, who's going to represent some of the academic interactions with biotech and industry for this discussion. And I've got a few questions to start things off, and then we'll open it up for comments from the rest of the people in the room. So the first question, and I'll just throw it out to anybody here on the panel, is what particular efforts within your research program are threatened due to current lack of funding?
Yeah, it's pretty clear. Just from the Kidney Cancer Research Consortium perspective, the infrastructure we have allows us to perform clinical trials that would not otherwise be performable without that infrastructure. It's not a large amount of money, but it's enabling in a way that I think is far greater than the dollar sum itself. Were that to go away, it would be much more difficult to do that. So I think we need that money to continue what I think is a pretty strong value proposition.
David?
Part of the reason I mentioned the story with IARC is, which was the center of the KIM-1 story, is that kind of research is probably the most at risk going forward, which is collaborating with folks who are outside the U.S. Essentially, we have an R01 that's focused on KIM-1, along with the folks at IARC like Paul Brennan and others, and we can't send them money anymore. So essentially, we can do research with them, but we can't move money from the U.S. It can't move towards them, and that's a big hit, and one that probably will last for a while. So even though the resources are there, we can't spend them, we can't collaborate, we can't take advantage of their biobank. It's a big threat to future research, not just on KIM-1.
Jim, do you have anything to add?
Well, I would say that, not sure. Okay. So we were fortunate to get a second SPORE in kidney cancer, and I think it would be great, to expand and beyond 2 SPOREs, getting a 3rd or 4th SPORE. And of course, the current climate makes it particularly challenging, but I think we should settle for 2.
Abhishek, do you have anything to add?
So I'll say a couple of things. So one is that we have noticed that from a basic science perspective, the trend that we have noticed is more and more the R1s, that are essentially flagships that keep the lights on in basic research labs, have begun reimbursement mechanisms, right? People are requesting to see more and more preliminary data, complete stories. The R1s have started resembling manuscripts. They are no longer exploratory, high-risk questions going after conceptual advances. They are essentially, we figured everything out. We're just going to write it in a way that will appear that there are some questions left outstanding and just give us the money so that we can pay for the next 5 years to do other stuff, right? This is not how we should be, in my mind, operating and doing science. So anytime a structure like that starts to collapse, we then immediately put a stop to new discovery because all the stuff that had to be paid out has already been done, right? We now stop doing any additional work at that point if the money stops coming. The second thing I'll bring, and this is not directly related to the amount of productivity, but rather I would really encourage people to think about the anxiety under which everybody is operating right now. This is not something that you can evaluate or measure very easily, but you're constantly worried that your money is going to run out as quickly as an email can be sent to your inbox. And this is a real problematic situation at the end of the day. So productivity is not just threatened by the fact that you don't have actual dollars in the bank, but also by the persistent fear that there's a sword hanging over your neck, which can come down at any point in time. So there are two aspects to both things.
Thanks.
So I'm mic'd and I can talk directly. I want to just add to David's point of view. I mean, since I was a resident and fellow, I had international collaboration. That's how I started. And these are threatened. We have an R01 with Maxine's son and a collaborator in the UK. And we had to reshuffle everything in the progress report to keep the money in the US. So I don't understand. I thought Canada is our 51st state, and suddenly it's not. So that's very important for the future. But to kidney cancer, I'm a bit with this climate worried because, you know, it doesn't mean that that breast cancer, prostate cancer are better, but they do have a diverse, just because how common they are, mechanism of funding through the BCRF, the Prostate Cancer Foundation. We don't have that. You've seen that with foundation overall in 1 year, less than $1 million was granted. So if I have a junior, a very young, undifferentiated Mike Atkins, Ari Beldegron, Abhishek, how can I get them, even if I provide free mentorship, to go to kidney cancer, right, David? It's going to be very challenging to do that. And I think, you know, you'll have way less with this. And other cancer will be affected, but to a lesser degree.
Thanks. So the second question I have is, what alternative funding sources have you tapped into to support your research? So Eric.
Yeah, I'll start. I mean, obviously, the clinical trial, the money that we get from the KCRP provides the infrastructure to be able to actually conduct these trials. The trials themselves, we have industry funding to pay for the trials per se. So, you know, in terms of what could we do in the future, it would be to, you know, depending increasingly on pharma to actually fund our program. But therein sort of lies, you know, the independence that we have in terms of being able to choose how we actually design our trials, and that is then impeded as you become more and more dependent on other organizations that may not have exactly the same alignment. So I think we have potential here for looking for other sources, but it won't allow us to continue developing our infrastructure as independent a manner as we are now.
So Jim, you're in Texas, so you have access to CPRIT.
Yeah.
Has that helped at all?
Yeah, it's been a big help. This is the Cancer Research and Prevention Institute. It is an initiative similar to what California did with the stem cell research, and it's obviously focused on cancer. I think they have about $300 million a year that they distribute for cancer research. I can tell you exactly how much of that is gone to kidney cancer, but it's not a trivial amount, and of course we are not the only beneficiaries, MD Anderson and other Texas institutions. And one could add to the list as well, obviously this is a small portion, but I think many of our institutions benefit also from wonderful patients and philanthropy, so that can be another source of funding.
David, do you have anything to add?
So over the years, as you know, we've gotten small amounts of funding for the SPORE program from the institution, which is obviously under some strain, from industry. We've asked them to indirectly fund some of the work, some of the translational work, for example. And when you have clinically annotated tissue, you can find funding sources. It's a little bit more easy. So having a SPORE tissue bank has been helpful in that regard. I think we've also gotten money in small amounts from patient advocacy groups, and that's the kind of thing that we could potentially build on. So for example, if we only can give 2 career enhancement awards per year, if, for example, KCAN or some other group, KCA, could fund a third, that would be helpful in both of our realms. So you can imagine ways in which we can build on the structures that currently exist. But I'm actually, I hate to say this because it's a somewhat negative session we're having here, but I'm actually somewhat hopeful about the future. And this may sound morbid, I don't want you to take it the wrong way, but it turns out that kidney cancer in general, and kidney cancer specifically, doesn't just affect one group. It doesn't have any political preference, at least as far as I know. And that the more we can amplify some stories, of how what we do impacts more patients than maybe come to see us directly, more we can talk about the impact we might have in rural areas, for example, not just in big cities. Like, here's a question for the group. What's the state in the country with the most quickly rising incidence of cancer? Does anyone know?
What? Repeat.
The state in the U.S. where the rate of cancer incidence is increasing the fastest. It's Iowa, or Iowa's certainly in the top 2. And so, with dramatic increases thought related to agriculture, nitrates, all that sort of stuff. So trying to study that, trying to understand, does that impact the incidence of kidney cancer, for example? Making those connections and then making an impact to try to improve them is a way we could move forward to get more support for our research, I think.
Tony?
Yeah, no, there is one thing also that, I'm lucky to be at an institution that actually it's in fabric with our founder, Dana-Farber, relied on philanthropy. He was the first guy, at least to my knowledge, to go on radio to bring, little boy Jimmy to talk about cancer, where my mother, for example, in a different country still doesn't tell her neighbors what her son does, the C word. So he started that, and that helped, and we have a lot of events, and in the richest country in the world, by far, the United States, with a lot of dispersed wealth, I have to say. For example, I do the Jimmy Fund Walk, I do the Pan Mass Challenge, and I dragged some of my colleagues based on the differential power, like Brad McGregor, Vincent, and now Stephanie, to do the Pan Mass Challenge, where we rise, we raise money, etc., and that goes back to this. That's another way, especially when the funding is low. So I want to talk about the power of philanthropy, a bit, and that helps from time to time.
So thanks. Abhishek, do you have anything to add?
I agree with everything that people have said before. The one, I think in terms of foundations, I think many of us rely on American Cancer Society funding, for example, a lot of money, especially for trainees as well as for high-risk projects. We get a lot. Much like the PMC, the Cleveland Clinic and the local community there has a Velosano bike, right? So we rely on a lot of philanthropy and support in that way. Sponsored research agreements with industry whenever possible. So I think folks have covered most of it, but we are all using the same additional tools and resources essentially to try and continue.
I mean, Brian mentioned this earlier, but probably the best thing we could do as a community is try to restore the funding level for CDMRP. And that, to me, gets at telling these stories about how kidney cancer affects people in different communities, also getting at the whole veterans angle of kidney cancer in people who are active duty, military or veterans. We all have those stories. We need to amplify those, I think, because that's the quickest way to us getting a large chunk of money on an annual basis, I think, to restore that $50 million.
So I have a few more questions, but I don't want to eat into the next session, but I'm going to open it up to people here if you have questions or comments you want to make.
Okay, all right. So I have two comments. One is that I have to say that the cornerstone of the or career development still the NCIR ones. So at section for a number of years, I realized one limitation for the field is that there are few kidney cancer researchers who is willing to serve in study sections, or actually study sections don't normally involve a lot of experts working kidney cancer on reviewing those kidney cancer grants. So I think that I would like to call that for those people who work on kidney cancer, you were asked by serving the study sections, review panels, please donate your time, because you guys are the experts that can review the grants in the most relevant way, can actually put in the most constructive comments, because oftentimes when I submit an R1 grant, I found some of the people who review the grants who has this irrelevant comments or something outside of the kidney cancer, obviously that you know that they don't appreciate the science. That's number one. Number two is that I would like to emphasize now single PRR1 is getting harder and harder. So I think that we should consider as a community to actually form the team to apply for some of the project grants, UO1, PO1 mechanisms that actually take advantage of complementary expertise from each individual investigators, either from the same institution or even from partner institutions, that will actually serve as additional ways that we can broaden this horizon in terms of grant fundings. So those are just two comments I have. Thank you.
Thank you for those comments. Anybody else have comments or questions for the panel? Okay, in the absence of other comments or discussion, Jim, you want to make a final comment?
So I'd like to add a couple of thoughts, at least for me, my concluding thoughts. So first of all, I think as a community, there is a lot to be proud of. When I started in this field 20 years ago, we had one drug approved by the FDA. Today we have over a dozen. Survival has gone from 10% in 5 years to over 50%. Our patients are doing much better. So I think, I don't want to end up in a sad note. I think things are challenging, but instead of feeling overwhelmed, I think we need to feel proud, and we need to think about what we're going to do to face up to this challenge. And what I would propose to the community is let's try to do that by working together. And what I mean by that is, I think as Chin just mentioned, we may be able to work together through PO1s or putting other grants together, but sharing reagents, and I can tell all of you, if there's anything that we have that you can help us turn, tumor grafts, cell lines, genomics that can help your research program, let us know, because we'll share them with you. So I think we may be able to do better by fostering the great network that we already have and continue to grow collaborations.
So I want to thank the panel and for a very interesting discussion and something that we can think about as a community as we go forward. Thank you very much.
Thank you, Dr. Atkins. Thank you, panel.